CRISPR, clustered regularly interspaced short palindromic repeats, is a revolutionary gene editing technology that has the potential to be used as a molecular surgery method. While it is primarily known for its applications in genetic engineering and research, CRISPR can also be harnessed as a tool for precise surgical interventions at the molecular level. CRISPR-based molecular surgery involves utilizing the CRISPR system to modify or correct genetic sequences within living organisms. The key components of CRISPR Cas9, the most commonly used variant of CRISPR, are the Cas9 protein and a guide RNA molecule. The guide RNA is designed to target a specific DNA sequence, while the Cas9 protein acts as a pair of molecular scissors that can cleave the DNA at the target site. In the context of molecular surgery, CRISPR can be used to correct genetic mutations responsible for various diseases. By designing a guide RNA that targets the specific mutation, scientists can deliver the CRISPR Cas9 system into the affected cells and initiate a precise cut at the mutation site. This cut triggers the cell's natural DNA repair mechanisms, which can be harnessed to introduce desired changes or corrections to the genetic sequence. This approach holds tremendous potential for treating genetic disorders by correcting the underlying genetic defects. Moreover, CRISPR-based molecular surgery can also be employed to introduce beneficial genetic modifications into cells or organisms. For example, researchers are exploring the use of CRISPR to enhance the expression of therapeutic proteins, introduce resistance to infectious diseases, or improve the overall health and function of cells. While the concept of using CRISPR as a molecular surgery method is promising, it is important to note that the technology is still under development and requires further refinement and rigorous testing before widespread clinical use can be realized. There are several challenges to address, such as ensuring precise targeting, minimizing off-target effects, and optimizing delivery methods. However, ongoing research and advancements in the field hold great potential for the future of CRISPR as a molecular surgery tool.